if all the delegates could please keep yourself on mute during the talks, so that the presenters can continue to present without interruption, that would be really great. But please do add in questions onto Slido, which we'll mention or will come on the screen now. So instead of using the Zoom kind of chat, if you kind of go to slido.com and enter that code or use the QR code, you should be able to add in all the questions and upvote other people's questions as well. And I'd encourage you, if possible, to keep your cameras on so that people, kind of the presenters, can see that there are people listening on the other end, and also come up with some really great questions for the presenters to show. And I think that's everything I need to say. Just check my list. So without further ado, I'll pass over to Elise, who'll take us from here.
Thanks, Josh. So the first talk we've got is quite interesting, at least for kind of looking at the implementation of in-house software development. So I'll pass over to Laura Smith.
Thank you. I'll just share my screen. Share. Just loading. Okay, hopefully everyone can see that. So hello, my name's Laura Smith. I work in radiotherapy physics at Southampton, and I'll be presenting today the development of a software verification and validation system for our in-house software called Artemis. So just a quick overview of what I'll be talking about, a little bit about myself, then about what Artemis is. I'm mainly focusing on our software testing system that we're in progress with, a quick roundup, and then what we're looking to work on in the future. So a little bit about me. I finished the STP in 2018. And since then, I've been working in radiotherapy physics at Southampton, mainly in the quality assurance team. So doing quality assurance and commissioning for linear accelerators. But I expressed an interest to be involved in some computing projects. And so since last year, I've been helping out with this software testing project. And yeah, so that's where the early career comes into it for me. So what is Artemis? Essentially, it's a web-based platform, and it acts as a DICOM hub, a centralized DICOM hub for our radiotherapy department to help improve the flow of data through the pathway. And this uses business process model notation. So you can see here from the animation, it's the flow from pre-treatment CT all the way through to the final export of the radiotherapy plan for treatment delivery. And I was first involved with this project testing the radiotherapy plan viewer module. So here you can see this module is for displaying CT scans and radiotherapy DICOM structure sets and dose cubes. And so I've been involved with testing this module and then branching out into developing a verification and validation system. So why do we need a testing system? Well, of course, we need to minimize the clinical risk of our in-house product. That's the main goal of this project. And to comply with all the current standards and guidance relating to software development. And also we want to strive for high quality user experience. So the users will be our clinical colleagues, and we want the software to be user-friendly and minimize the risk of using it incorrectly. So we want an intuitive software for everyone to use. So how did we do this? We've reviewed the IPEM best practice guidance and the relevant standards for risk management, which I'm sure you're aware of. So just a few listed here, including the NHS Digital Clinical Risk Management Standards, and also briefly reviewed the software testing standard, the ISO 29119, and reviewing these to get us going on starting our system. But in-house development does present fresh challenges. It's a very fast-paced development of the software. There's new features and bugs being fixed all the time. So it is quite challenging to keep up with the testing. And also the project has been in progress since 2022. So there's quite a lot of pressure from clinical staff to, when we roll this out, what they're expecting it to be. And also working in-house, the development team are very flexible to requests from clinical users, and therefore we need our testing system to be just as resilient and robust to be able to cope with this fast pace. So what did our radiotherapy SciComm team look like not too long ago? Well, the SciComm team is made up of 5 colleagues, 2 clinical scientists, a computer technician, and 2 software engineers. And this is how I sort of see the agile methodology working before I sort of joined the project. So starting with user requirements, working through the software requirements into the planning and design phase. So they have fortnightly sprints and work through this cycle of developing and unit testing the software and the new features, and deploying to a mock system. So the mock is the testing environment, and this is completely separate to the clinical environment of the software. And then this cycle continues until they're happy to release to clinical. But of course, best practice advises for the development and testing to be independent. So the first step to the software testing system was to create these independent teams. And that's where I've been involved here. So we've now got a well-defined development team and a well-defined testing team. And this removes any bias from the developers testing the code, and also with clinical scientists being involved in the testing team, it brings sort of clinical experience and maybe a fresh look at the software to try and test it in a way that the users will use it. So after deploying to the mock system, we then run our testing after this, and we'll continue this loop of sprints for each milestone until we're happy to release it to clinical. So the testing methodology, I have to admit, when I first started, it was very overwhelming. There's a lot of different types of testing that I'd not heard of before. So initially, I think the fast pace and all the different terms that were quite new to me, it was very daunting to sort of get involved in such a big project. But after working with the team for a while and reviewing all the guidance, it soon started to make sense on how these different tests take part in the system. So you can see here Venn diagrams sort of can organize them into 2 main areas. You've got verification testing and validation testing. So verification is very much on the technical side. Am I building the product right according to the software requirements? And then moving through to the validation, so more the user end. Am I building the right product for the end users? But our testing team, we're not full-time trained testers. We don't have a lot of dedicated time to spend on the testing. We have other clinical duties to carry out, and therefore we've taken a risk-based testing approach. Of course, that's recommended in the standards, including the software testing standard. So we focus on the most important features that carry the highest risk going forward, and prioritize our testing in this way. So after working on this project for a few months now, I've learned that you really need to think like a tester. You need to think outside the box, and it's quite different to the quality assurance I've been used to. You need to think in a very different way. So here I've just, as a snapshot, tried to capture how our software testing cycle is going. So we base it around a weekly sprint. So after reviewing the software requirements, we have come up with a defined expected behavior of our software, which we can test against. And we're continuously developing test cases, with the majority of our time focusing on performing and documenting the tests. Reporting any defects that we find, and reviewing these on a weekly basis until we're happy to release the software. So how do we report our defects? We're using Atlassian Jira. and this is working really well for our team. We've come up with quite a nice system for the flow between development team and testing team, using the priority and status filters. And this is something that we'll review at the sprints, and change the priority based on where the roadmap is going. So just in a little bit more detail, one of our issues, and you can see here on the left-hand side, our description, we've got a well-defined method for reporting our defects to make sure the development team can reproduce these, which environment we're using, and then what the actual behavior is compared to our expected behavior. And on the right here, you can just see a history of one of the tickets, going back and forth between the development and testing team. So we've got all of these documented. Then on to our testing review. So like I said, we have a weekly meeting just to review any changes to the software. Have there been any bug fixes or new features released? And then we'll review the clinical safety hazard log to make sure there's nothing we need to add or update on there. And also look at our documentation. So documentation is something that is we're working on at the minute. And essentially, we're trying to ensure traceability through from all of our software tests all the way back through to the software requirement specification. So just an example, we've got our software test plan that we're continuously adding to. We've developed a software test case repository, and on the right here you can see an example of one of our test case descriptions. So this should give enough information for someone to repeat this test at a later date using the test case ID and the test ID, which hopefully will be traceable back to the requirement. So going back to our Venn diagram of validation and verification, there's one main thing missing here, which is the user acceptance testing. So as another sort of step in our testing method, we've recently introduced a new staging environment, which sits between the mock and the clinical. So this is sort of a more stable release of the mock system. And we've now got a clinical commissioning team, which is made up of 3 clinical scientists completely external to the SciComm team. So if we come back to our cycle here, after testing within our testing team within the SciComm, we'll deploy then to staging once we've signed it off and we're happy. And the CCT can do their own clinical testing and feedback to us with any improvements or bugs that they've found. We'll repeat this cycle until finally they're happy for it to be released clinically. So in a nutshell, what have I learned from this project? I'd say it's been a steep learning curve. It's a very kind of fast-paced environment, and it's very different to what I've been used to with QA. And I'd say time management and organisation is key. And it's actually kind of, I've realised that software testing is completely its own profession. And we're actually sending the software testing team for international software testing qualification certification to make sure we've got the proper training. And after this, we'll probably, develop our software testing system even further. And what could be improved in the future when the development slows down a bit and it's a bit more stable? Hopefully, we can have a more rigid deployment and release schedule that we can organize our testing. And if we could automate any documentation, that would be amazing. And in the future, we hope to automate a lot of the regression testing, because this does take a lot of time. So if that could be automated, we could improve our test coverage and test a few more features that we currently don't really have time to do. And this would be using systems such as X-Ray, Jenkins, Postman, and Cypress. And yeah, we're also developing a minimum test set for routine self checks and QA. And we're continuously sort of working on our documentation, as I mentioned earlier. And that's it. Just like to say a massive thank you for all of the SciComm Artemis team. Has anyone got any questions? Thank you.
That was really great, Laura. It's interesting to kind of see how different scientific computing departments are set up and kind of how it's been implemented in Southampton. So we've got a slide which I think should be brought up soon by the IPEM. But if you guys have got it up there we go. Okay. So we'll go for the first one. Do you have any resource challenges kind of regarding this in terms of the develop test split?
Do the teams always test develop or do they swap? So yeah, they're very sort of defined teams. We don't swap anything. Do you have resource challenges? I mean, there is a lot of testing to do. So in terms of covering all of the tests, yeah, we do have challenges in that sense, but that's why we're choosing this risk-based approach. Yeah, I'm not sure.
Yeah, that sounds great.
Yeah.
I mean, it's quite different from my department. I think it's interesting having that split to kind of see, you know, sometimes it's better to have someone else who's not involved in it, have a fresh pair of eyes in terms of testing. Often they'll pick up things which you wouldn't necessarily pick up. So the next one is in terms of the different standards. So how have you used the ICE 62366 within your quality testing system?
I'm not sure if that's. I think we've reviewed it as a department, but specifically, yes, yeah. We're not strictly a medical device, so we're sort of adhering to the standards for best practice, but we're not working towards that specifically. We've been using it, all the standards, as like a guidance, essentially.
How have you found kind of working towards the standards in terms of getting the quality system in place to meet them?
I mean, it's still a work in progress, the quality management system. So with the development being so sort of pressing, most of the resources is going on the development of the software right now, and quality management system has been in development alongside it, but it's it's quite challenging to, you know, focus on both of them at the same time.
Yeah, I mean, in terms of that, kind of thinking about the kind of capacity of the different sites, how do you find it with kind of your software testing role as well as the role that you do for radiotherapy?
Yeah, it is quite challenging, I'd say. Sort of, like I said, it's almost like putting a different brain on. So yeah, with the team and just communicating with that, what sort of timeframes we're aiming for, and managing to slot it in like that is yeah, how we're managing it really at the minute.
That sounds great. Yes, I know from experience for me, it's difficult to fit all the kind of all your different hats on, as you could say, for the different jobs that you do. So you mentioned about how you're quite kind of integrated with the clinical team and getting feedback and things. Obviously, with the agile system that you guys use, how do you find kind of dealing with the requirements? Is there any kind of creep within the mock system that you guys set up?
How do you prevent? So you mean how do you prevent requirements creep into the mock system? Well, we're sort of constantly reviewing the requirements as we're developing the software. So if we are sort of taking requests, then we'll review the requirements alongside management as well to see whether that's the way we want to go. So yeah, it's in terms of the development side, the requirements are sort of met as they're seen needed, and we might put them in the backlog to implement at a later date once we've got the sort of essential needs met.
So for the next question, how do you define the test cases? Is there any particular methodology that you use?
This is an area that we're sort of learning about still. Like I said, we're going on training soon, so we don't have a specific methodology for defining them at the minute. We're reviewing our expected behaviour and just sitting down and working through the modules and just trying to sort of come up with all the different tests that we can imagine. But we don't have a specific methodology currently for defining these tests. So hopefully that's something we'll learn at the training.
Yeah, that sounds great. So we'll go for the final question. Are you just developing and testing on a single application, or do you work on multiple applications?
This is I'm just working on this specific project. So this is the main software that we're developing in-house at the minute. Maybe in the future there'll be other ones, but it's just a single application at the minute.
Well, that sounds great. Thank you so much for your talk. It was really interesting. I definitely enjoyed it. It's always good to see how other departments work. So we're going to go on to the second talk now, and this is by Jonathan and James from South Devon NHS Trust, and it's called a data warehouse solution for setting LDRLs. So over to you guys.
Thank you very much. Thank you. Yeah, you want to go? Let me just make it on. Can everyone see our screen okay?
Sorry.
So good afternoon, everyone. I'm James. I'm a first year STP trainee working in Torbay Hospital. I'm here to present with my colleague, Jonathan. Hello, I'm Jonathan. I've been working as a medical physics technician for a year. I'm a recent physics graduate, and I'm going on to do the radiotherapy STP at Clatterbridge in September. Okay, so before we dive into the meat of things, so a bit of structure for you guys for our presentation. We're first going to introduce the idea of diagnostic reference levels, DRLs. We're going to talk about the local challenges that we face locally in setting these DRLs, and then our project brief. I'm then going to hand off to Jonathan to talk about our results, impact, and subsequent investigation from resulting from our project. So patient exposures must be a LARP, but how do we determine this? So diagnostic reference levels are statistical quantities obtained from a distribution of diagnostic exposures representing a population. So DRLs can be used as a landmark to gauge what is good and normal practice versus what is bad and abnormal practice. When we talk about DRLs, we normally talk about two types, so local diagnostic reference levels, LDRLs, which is usually defined as the median of the distribution of appropriate dose quantities observed at a healthcare facility. And then when you collect the LDRLs across a country or a nation and take the 75th percentile, you obtain the NDRL, the National Diagnostic Reference Level. So setting LDRLs locally involved asking radiographers to record the data on a piece of paper and then digitizing that into Excel. Or alternatively, we can request data from CRIS, which has its own issues, and we will touch on that later on. Previously, before our work, an MPE inspected each data set and calculated LDRL. Now, this is advantageous because there's a wealth of clinical input into setting those LDRLs from the MPE, but it's because there's no formal method that we have it can vary quite a bit, and it's also work intensive and time consuming, which is not sustainable. So LDRLs are a requirement of IRMA, and a CQC inspection indicated that we didn't have a formal methodology in place for calculating LDRLs, and there was not a standard review or audit process we had in place. It also found that we had there was a poor understanding from the clinical team on what DRLs are and how we use DRLs to ensure doses are alarmed. So the local challenges are because LDRLs are statistical quantities, you need to process quite a lot of data to get statistical certainty. Now, processing those data manually is time intensive and would be detrimental to an already stretched physics workforce. The process we had previously was quite sporadic and only took account a small subset of patient data. The other problem we had was that the data that we processed needs to be recorded consistently and accurately. And we'll again touch on that a little bit later on. So our project brief was quite simple, is to determine a standard operating procedure for setting LDRLs, and then to create a method to automate or semi-automate calculating LDRLs. And from those LDRLs that we've calculated, generate a report and compare them to national diagnostic reference levels. So this is kind of the flowchart of what we've been doing. So on the left side, where there's different image modalities, those informations gets loaded into CRIS. And this is essentially the kind of left-hand side of this diagram. Our main bit of work is into how we query that database and then how we generate report, which then feeds into projects. We've done this by scripting a query. Essentially what it does is go through the database and segregate data, and then goes through an iterative process to challenge whether the data's got good enough quality and quantity to set an LDRL with good statistical certainty. To just validate what we did, we took LDRLs for a CT room that was set by an MPE, and we fed the same data to our script. And it shows that there's quite good agreement between what we've set and what our calculator is outputting and what has previously been set. So we're in the right ballpark, but we are expecting subtle changes because of how we've standardized calculating these quantities. So for example, in virtual colonoscopy and neck thorax abdo pelvis, there wasn't, the calculator decided that there wasn't enough data to set those quantities. I'm now going to pass over to Jonathan.
Thank you. So I took over from James at the start of this year on this project. Because of course we're using CRIS data now, the complete CRIS data set which we pull, we are limited by the quality of the data that we receive from CRIS, and we can split this up into quantifiable issues and unquantifiable issues. So first we'll have a look at the quantifiable issues. So for the CT modality, we're looking at 77% of the exams that this trust performs annually have no weight recorded, and our methodology requires that we have weight dependency. So we have LDRL selected from the median of 50 to 90 kilogram patients, according to recommendations. So this means that 21% of the exams that we pull from CRIS can calculate an LDRL. For plain film, it's a little more concerning. We're looking at 99% of the exams that are performed. Radiographers currently sum all projections into all. So there's no way of seeing from CRIS data alone what these projections were retrospectively, which makes it quite challenging. And the only way to get around this is by reading DICOM headers directly. More than 99% of exams have no weight recording. Clinical teams keep reporting this is because the scales keep breaking. But these are issues that we were not aware about before. Fluoroscopy, we only have 2 out of the roughly 5,000 exams we perform annually having a weight recorded, and there's no unit recorded for 44% of exams. So this has all been highlighted by our work. In terms of unquantifiable issues, we don't know the full extent of how these affect our data because it's quite hard to determine, but radiographers have reported that this occurs. So often the room will be recorded incorrectly. So you have a difference between the room that the patient was scheduled to have an exam or what have you on, and then when they, on the day, they end up in a different room all of a sudden. But unfortunately, one of the old room will be recorded. CRIS codes are often used incorrectly. Sometimes we have multiple exposures summated onto one CRIS code. Often in fluoroscopy, an exam will be started on an interventional CRIS code and then summated later. They'll later have an interventional procedure, but that will all be summated onto the original diagnostic CRIS code and reported then, which then means that has a hugely inflated dose. The dose quantity units in fluoroscopy are often recorded incorrectly. So we have a variety of machines and different equipment throughout fluoroscopy, and these report the dosage, but they deliver differently. Some report it in gray, some report it in centigrade, decigrade, and we require currently radiographers to convert that manually into centigrade centimeters squared, which has created some challenges of inconsistencies in the data. Many of these issues are down to training. These can be negated by directly reading from DICOM tags, but at the moment, we haven't had the resources to do that just yet. So the immediate impact of our work was we were able to produce LDRLs with consistent methodology documentation, which are an accurate reflection of the large data set that we're now putting in rather than the subsets that we were before. We now have the ability to launch investigations to ensure that patient dose is a LARP and the quality of exams is sufficient. So we launch investigations when the LDRL or room median dose exceeds NDRLs or is significantly below, because that raises questions on whether the scans have sufficient quality, image quality. We also raise it when we have significant differences in median dose of the same exam across different rooms. So that raises questions about whether the different equipment was performing optimally, which before we had no procedure in place for. So we've been able to reduce the physics workload significantly, and we've established formal methodology for both producing LDRLs annually and also auditing them. And this is ensuring compliance with IRMA and is fulfilling the CQC objectives that we were given as a trust. So our first investigation was into C-spines. So the local LDRL was observed to be 121% the national DRL. The national DRL, of course, being at the 75th percentile and the local DRL being at the median. So we can kind of, ICRP recommends that we treat the NDRL as a upper limit. So we initiated an investigation. Physics removed the high-res presets, which were not deemed to be required. And they investigated the Scout. So we have some GE scanners. We have 2 GEs and a Canon. The set and Scout on the GE is used to determine MA and KV modulation. And this was LAT, but it's been changed to AP. So you're looking at a narrower length rather than a wider one. And this did create some streak artifacts. So the field of view was minimized to avoid this. Radiographers focused on training. They iterated that patients had to be moved to isocenter before scans were performed. And it was requested that patients are asked to lower their shoulders with their arms down to make sure they're not instant to the beam to reduce the dosage. This reduced the LDRL to 97% whilst maintaining diagnostic quality. It's still a little bit high. We believe this is because the NDRL was gathered from a lot of trusts that weren't scanning down to T4 for CT trauma spines, which we do. So we're expecting the NDRL to increase as this practice is carried out more routinely across the nation. This year then, we investigated CT abdo-pelvis. We observed that the LDRL was 160% of the NDRL, It turns out that this was actually a bit of a red herring. We investigated this and we discovered that the CRIS code was being used incorrectly. There's a summation of multiple acquisitions into one procedure often. We managed to revise the LDRL using our methodology to 70% once we got radiographers to go through the data in packs. We also identified that there was poor practice in recording weights, especially for this, and this was leading to a higher LDRL. Many patients who weigh more than 90 kilograms were being considered as weighing less, and thus they were included in our methodology, and so the LDRL median was increasing. So this demonstrated to clinical staff the importance of recording weight consistently and accurately. Recently, we've launched an investigation into CT heads. So we have two types of scanners, as I mentioned. We have a Canon and two GEs. Both scanners at the moment are producing diagnostic images. You may be able to see that there's slightly higher scatter on the GEs, but we did note that there was a lower median dose on the GEs. In fact, the dose was so low that it was below our local investigation levels for this machine, so which we set at the 25th percentile of trust data, and it was also significantly below the NDRL. So this means that we could currently be missing diagnostically relevant information on the scans we perform, which is a bit concerning. And this, of course, was something that we wouldn't have noted before necessarily. So a project was initiated, and this has had region-wide outreach. Registrars from across the peninsula are being contacted to help us assess the quality of our images. We have about 60 images that have been selected by physics and radiographers to assess them for their diagnostic, whether they're diagnostically sufficient quality. And we're relying on expertise from various centres across the peninsula as well to help us do this, because this is really a flagship project for us. We've never done something like this. So to summarise what we've done and the work ongoing, we're limited by the quality and depth of data from CRIS. Staff training needs to be improved. Governance surrounding this can work on that. But of course, there are workload pressures for clinical teams, and they've expressed that they're not overly keen on having to record everything extremely accurately because this will reduce patient workflow. So we're looking towards another solution going forward, but this has acted as a stopgap, as it were. We've established SOPs for calculating LDRLs where previously these didn't exist. We've established processes for auditing LDRLs and median doses across rooms. We've been able to launch investigations, and this is all helping working towards ensuring patient doses are up. We've managed to create and complete image optimization projects. But looking forward, we are hoping to move on to a more formal dose management software solution. We've considered OpenREM, but IT challenges locally mean that developing open source software is quite tricky for us, especially as there isn't much software expertise in the department. And we're also investigating a proprietary solution, hoping to link with trusts across the region, and hope that that will provide more accurate data with deeper insights. We're very grateful to the help we've received from across radiology at Torbay, specifically Kate, Catherine, Becky, Sharon and Katie. We're very grateful, and this project wouldn't have been possible without them. And we thank you very much for your attention and open the floor to any questions.
That was really great, guys. It's quite interesting to kind of learn about different departments. I haven't got a radiology kind of background myself, so it's always nice to learn about something new. So going on to the questions, first one is, are the data limitations fed back to try and improve the process?
So yes, we have an image optimization group that's been formed since the CQC inspection. And I have presented to a couple of times to them to basically say this data is not of sufficient quality for compliance with going forward. The guidance does say that as long as going forward you produce more and more LDRLs, that's fine. So we're in a better place than we were. But yes, so we do feedback regularly to radiology as a whole on where we're getting with this and what we need to work on.
Sounds great. So are the patients who do have the weight information representative of the whole population, would you say?
So we took advice from the ICRP and IPEM guidelines, which is to take a median on essentially a particular weight range, which I think is 50 to 90 kilos. So when we separate out our data, we look for that. We do look for that specific weight range, but we also calculate LDRLs for other weight range, so below and above that.
I think it's important to bear in mind as well, the NDRLs are calculated for the same weight ranges. So in order to compare directly with them, it's important that we have methodology that's consistent. But of course, we don't have weight data, as you can see, of outstanding quality. So it's difficult to say what the weight distribution locally would look like.
So kind of considering the issues with the weight data, how did you deal with that? Did you do any kind of like augmentation, or did you look, oh, okay, there's a bias to one side, or is it just kind of, you have what you have?
So if there weren't sufficient patients who had an exam that was weight recorded, we would provide a LDRL that wasn't filtered for weight. We did do a regression between weight and dose delivered, but with the uncertainty in weight and dose, especially in modalities that aren't CT, it was really quite tricky to get much from this. So at this stage, we're just sort of putting it in a bracket and then calculating the median from that, which should, using the median, it should mean that we try and exclude outliers.
Sounds great. So is the data warehouse the CRIS database, or are you extracting the CRIS data into another database?
Yeah, so we actually export data from CRIS. So I think that's the ETL process that I was describing. So we extract it from CRIS and then transform it into something that that is useful for us, and then we load it into our scripts.
Yeah, we just get a big CSV file, and from there, and then we apply lots of filters and criteria from it based on ND guidance.
Great. And then for the final question, is your methodology generalized enough for future uses?
So I have taken on the development most recently. James worked on this a year ago, which is why he took the first half of the presentation. And locally, we do struggle with coding experience. So a priority for me has been developing it in a way that's future-proof and has a GUI that's very understandable and interactive and easy to use. In terms of new methods, I don't think at the moment it will be possible without coding experience. I've built it in such a way that it's class-based and it's object-based, based on a standard procedure, and then different modalities have sort of inheritances on top of that. So stuff like you have your standard methodology for CT, and then for plain film, you have to consider projections as well. So you also add that on top. So it should be quite easy for an experienced person to develop, and it should be fairly robust. But yes, it is important that going forward. It won't be supported, but we're hoping there'll be a dose management system before then.
So that all sounds great. It was a really interesting presentation, and seems like it was a very good project in terms that it highlighted kind of the information that wasn't there and should be recorded. So it's really great, guys. So moving on to the next presentation, this is by Bethan Morris from Leeds Teaching Hospitals NHS Trust, and it's called In Silico Modelling of Glioblastoma Invasion to AIM Radiotherapy Target Delineation. So I'll pass over to you, Bethan.
Great, thank you. So I'll just share my screen quickly. Okay, so hopefully everyone can see my screen okay. Great. Okay, so I'm Bethan. So I'm a third year STP trainee training at Leeds in the CSC group. So I'm in the clinical bioinformatics physical sciences specialism, which is the old version of the CSC specialism. So the work I'm talking about today is a project I did for my MSc research project as part of my STP training. So the work was entitled In Silico Modelling of Glioblastoma Invasion to Aid Radiotherapy Target Delineation. So I'll start with a little bit of background. So glioblastomas, or GBMs, are a particularly aggressive form of brain cancer. So they're the most common primary brain tumour in adults, and patients typically survive for just over a year with these tumours, so they're very poor survival times. So a challenge with treating these tumours is the fact that they are very invasive. So a typical GBM is sort of composed of a fairly solid sort of tumour core, and then surrounding that is an invasive margin. So it's sort of described as you've got your solid tumour core, and then individual cells tend to detach from that core and invade into the surrounding healthy brain tissue. And this invasive margin isn't visualised through imaging. So on MRI scans, we can only really see the solid tumour core part of the tumour. So to treat these tumours, surgery is often the first kind of line of treatment. So the surgeon will go in and remove as much of the tumour as they can, but they're typically only able to remove the sort of solid tumour core, and generally that the cells in that invasive margin are left behind. So currently, to try and treat these remaining cells, surgery is followed up by radiotherapy and chemotherapy. And unfortunately, because we can't visualize these invading cells, it's the way the radiotherapy target volumes are defined is just by defining a 2 to 2.5 isotropic margin around from where the tumor was originally removed to try and capture where those cells might be. But unfortunately, these tumors always recur, and typically they recur within that region where the radiotherapy was delivered to. So clearly there's some improvements that need to be made around treating these tumours to try and get better outcomes for patients. So the idea is that we could possibly look at defining new radiotherapy target volumes and treatments based on where those invading tumour cells are more likely to be, rather than just guessing by adding this sort of margin on. So the idea is to develop a predictive model of GBM cell invasion. So although we don't know where these tumour cells are, we do know some things about how they invade into the surrounding healthy brain tissue. The idea is to incorporate this knowledge into a predictive model. And we wanted our model to be driven only by routinely clinically available data. And that's because we want it to potentially be able to be translated into the clinic in the future, like a long way down the road. But yeah, so so tumor, if you've got your solid part of the tumour, and then individual tumour cells tend to detach from this tumour and invade into the surrounding healthy brain tissue. So in our model, we've decided to describe this by modelling this as by describing a population of GBM cells independently following a random walk to invade the surrounding healthy brain tissue. And in our random walk model, the steps taken by the cells are influenced by two main factors. So this is the brain anatomy and those known invasive behaviours of GBM cells. So first, if we talk about the brain anatomy, so we know that the brain is quite a complex structure and it influences how these tumour cells move throughout the brain. So the way we incorporate this into the model is by generating tissue masks from individual patients' diagnostic MRI scans. So we take their scans before they've had any treatments, and they go through some data pre-processing to generate these tissue masks, which are generated using an automatic segmentation algorithm. So this is an example of a tissue mask over here. So we have 4 tissue types that we're interested in the tissue mask. So the grey sort of region is the solid tumour core, and then the blue is the white matter of the brain. The green is the grey matter of the brain. The yellow is the cerebral spinal fluid, and then obviously the black around that is classified as not brain. So in our model, we restrict cell movement into the cerebral spinal fluid, the tumour core, and outside the brain. And that's because these tumours are rarely seed into the cerebral spinal fluid and outside the brain, so we don't allow that to be a possibility on the model. And we're also assuming that they're invading away from that solid tumour core because they're described as detaching from it, and we know they go into the surrounding healthy brain tissue, so we don't allow those cells to move back in to the tumour core. And then also we know that GBM cells move more easily through the white matter of the brain than the grey matter. So the way that we reflect this in the model is by allowing cells following the random walk to take a larger step size when they're in white matter than when they're in grey matter. So the other known invasive behaviours of these cells that we incorporate into the model we incorporate by including 3 different terms. So the first is a momentum bias term. So there is cell tracking data that shows that GBM cells move with some directionality, so they don't tend to just randomly move in random directions. There's some kind of general direction to their movement. So in the model, we assume that cells are more likely to step in a similar direction to the previous direction of movement. And then we also include a history bias term. So there are limited resources within the brain, and therefore cells are more likely to move to new locations where there are more resources. So in the model, we assume that cells are less likely to step into voxels that they have previously visited. And then finally, we incorporate a white matter bias term. So it's known that GBM cells migrate preferentially along the white matter tracts of the brain. So in the model, we assume that cells are more likely to step in directions where there is more white matter in front of them. So our biased random walk is off lattice, and cells are allowed to move through the space defined by the tissue mask for individual patients. So each time a cell following the random walk goes to take a step, we generate a list of possible cells a list of possible steps for that cell to take, and then we take a weighted sample from that list. So these sample weights are calculated using those 3 bias terms that I talked about in the previous slide. So the momentum bias, the history bias, and the white matter bias all influence which of those steps the cell is more likely to take. So we use this model to generate these predicted invasion maps for individual patients. So we basically simulate a large number of cells following the random walk, and each of these cells is initialized from a randomly sampled point at the edge of the tumour core, and then they follow the walk into the surrounding healthy brain tissue for a given number of steps. So the number of steps that we allow the cells to walk is sampled from a distribution to kind of reflect the fact that these tumour cells will have been invading for different amounts of time into the surrounding healthy brain tissue. So once we've done all those simulations for those cells, we basically count the number of cells that pass through each voxel and normalise those counts. So we produce this heat map showing voxels that have been visited by the greatest number of tumour cells to kind of reflect voxels that have had the most invasion to them, basically. And then we're able to convert these maps into RT dose files and then load them into the radiotherapy planning system that we use at Leeds, which is RayStation, so that clinicians can view these maps overlaid on patient scans. So that was a bit about the sort of model development. And then following that, we did sort of a preliminary clinical evaluation. So we produced predicted invasion maps for 4 patients using some example model parameters. And we asked 2 clinicians to sort of review these maps and evaluate whether some key invasive behaviours were followed correctly by the model. So these were, the first of these were no invasion across known barriers to invasion. So there are 2 sort of key structures in the brain that we were looking at. These are the falx cerebri, which is highlighted by this red contour here, and the tentorium cerebelli, which is highlighted by this blue contour here. So these are 2 structures in the brain that we know that tumour cells can't cross. So our predictive invasion maps ideally shouldn't show tumour cells crossing these barriers. But you can see hopefully on this example map here that these are being violated in both cases, because we've got invasion predicted down crossing this blue barrier down here and across the red barrier here. But the second criteria we were looking at were no invasion into the ventricles, and then the third criteria was increased invasion in the direction of white matter tracts. So I'll show you a couple more example maps here. So for this patient here, we've got the ventricles of the brain here, and there's no invasion into those ventricles, which is good. So that's predicted correctly. And we do see some increased invasion in the direction of some white matter tracts. There's some white matter tracts going up in this direction. And there's also a big white matter tract here going across into the other hemisphere of the brain. And we do see increased invasion in that direction. The falx cerebri is up here, and we don't actually see any invasion across that, but it could be that the tumour cells just haven't reached that far. But yeah, so some of the criteria have been, well, all of the criteria have been followed correctly in this map here. And then this one over here, we've got the tentorium cerebelli down here, and we do see lots of invasion going across that, so that's not being followed correctly in this example. But we do see that there's a lot of invasion going vertically, and this is expected because there are big white matter tracts that span the left and right cerebral hemispheres vertically, so it's expected to see a lot of invasion in this direction. So this was, the reviewers noted that this was quite a positive aspect of the prediction. So overall the clinical reviewers deemed that the maps were kind of broadly sensible, and they reflected some of the known behaviours of GBM invasion well and others less well. So the main sort of drawback that they highlighted was that invasion was predicted across impassable barriers in some cases. So that the main problem was the falx cerebri and the tentorium cerebelli. And I think the main problem with this came from the automatic tissue segmentation algorithm that was used, that it just wasn't picking up these structures, and they were just being, meaning that cells were just allowed to cross them. But on the positive, we observed that there was more invasion predicted to occur in a vertical direction rather than laterally. And this is because kind of 2 main reasons. So there are lots of barriers preventing lateral invasion. So you've got the ventricles of the brain and the falx cerebri sort of inhibiting that lateral invasion. But even where there was space to invade laterally, we still saw that there was more invasion vertically. And again, the reviewers noted that this was because of the presence of large white matter tracts that span the left and right cerebral hemispheres vertically. So they would expect to see more invasion vertically than laterally because of that. So that's kind of broadly my MSc project in a nutshell. So we developed a model of GBM invasion driven only by routinely clinically available data. And we produced some example predictive invasion maps for a small number of patients. So the reviewers said the results were initial results were promising, but there were definitely some issues with them that we need to work on. So yeah, there's definitely more work to do. So we need to do a bit of a sensitivity analysis to see how the model parameters change the maps that we produce. So those maps were just produced for some example modeled parameters. So we do need to do a bit more parameter exploration, and also to try and parameterize the model. And then we also need to look at validating the model predictions, which is going to be quite a challenge because of the limitations of data that we've got, and also the fact that these patients have been undergoing treatment. So there are all those influences to consider as well. And then also to consider how to address the challenges with the segmentation of those two barriers, which were definitely a drawback to the predictions. But yes, so thank you for listening, and I'm happy to take any questions. Thank you. Stop sharing.
That was great, Bethan. I think, yeah, it seems like a really cool MSc project. I imagine you found it really interesting. So going on to some of the questions, this one is, how did you collaborate with the radiotherapy team?
So two of my supervisors for the MSc project work within radiotherapy. So one is technically in the CSC team now, but he was a previous radiotherapy physicist. And then we also had the head of radiotherapy imaging was on my supervisory team. He was we had monthly project meetings where we would discuss the project. And then also one of the supervisors was a clinical oncologist. So she prescribes radiotherapy treatments to patients. So she was really giving her clinician's perspective on it as well. So the project was kind of suggested by, so I'm from a maths background and I did my PhD looking at GBMs. So I already had an interest in GBMs, and the department already had an interest in GBMs as well. So we once we sort of realised we had the same interest, that's kind of where the project came from. So there had been some previous work looking at doing a random walk model for sort of from a radiotherapy perspective, planning perspective. So yeah, that's kind of where I came in. So my role was I did all the coding and model development and yeah, most of the work, and then we had these monthly review meetings to sort of discuss. Yeah.
Sounds great. I'm also from a maths background, so
great.
great to see other people. So for the next question, what platform did you use to model in? And did you have to use any particular hardware or anything to run the simulations?
Yeah, so all the code was written in Python, and it was quite slow to run the simulations. So we ended up parallelizing the process across 10 cores. And so for each map is produced using 250,000 simulations of cells. So across the 10 cores, it still took about 4 hours to produce each map. So that's kind of the reason why I haven't got a lot of maps for a lot of patients, and I didn't get to do as much exploration of the model parameters as I wanted to, because yeah, it just took a long time to produce each of the maps.
Yeah, I can imagine. Previous modeling things that I've done have taken a very long time, unfortunately. So for the next question, how did you choose the amount of bias that was introduced to the random walk model?
Yeah, so this is one of the so the model parameters that I've talked about, those are controlled by those control the kind of strength of the biases. So I've just chosen some kind of middle-y values at the moment. So there are kind of specified ranges that those parameters can be, and I've just kind of chosen yeah, kind of intermediate sort of values to produce those sort of example maps as a sort of initial pass. But that is something that needs to be sort of tuned, looking at more patient data. And yeah, I need to, in the sensitivity analysis, I need to see how much the maps change when I change the strength of those biases. So yeah, it's definitely more work that needs doing on that.
Great. So we'll do one more question. What automatic segmentation algorithm did you use to create your tissue masks?
So the automatic segmentation, I didn't actually do that part. So that was done by one of the radiology registrars who very kindly helped us out with that. So I don't know the full details of that, but if you're interested, I can definitely point you to his paper where he talks about it. But yes, so I don't know off the top of my head, sorry.
No worries at all. Yeah, that was really great. I really enjoyed it. It's nice to see kind of what everyone does for their different MSc projects. There was quite a range in my year as well. So always interesting to see the different work. So we're going to go on to the panel discussion, and I'm going to hand over to Josh, who will be leading it.
Great, thanks very much, Elise. There were three really fascinating talks there of quite a wide range of topics, which I really enjoyed. So thanks very much to all those speakers there. So this panel discussion is going to include Helen, Isidora, and Elise, who all have kind of recently qualified as kind of in the CSC kind of remit. So we're just going to ask a few questions to them of their experiences as they've kind of progressed into this career path, a little bit of how they got here, and then as well there's an opportunity for, if anyone in the audience to ask questions as well, again pop them into Slido and we'll see what we can get through before the end of the webinar. So great. What we'll start with then, I think, is just a chance for you to introduce yourself. Where do you work, and how long have you worked in this kind of CSC environment? Helen, why don't we start with you?
No problem. So yes, my name is Helen Lasware. I work in the scientific computing department at the Royal Surrey in Guildford. And like a few other people here, I did the scientist training program. So I started in 2019 and finished that in 2022, and have since gone on to a job as a computer scientist within the same department that I trained in. So I've been there now for 5 years.
Great, thanks very much. And Isidora, how about yourself?
So, hi, my name's Isidora. I work in the Radiation Physics and Radiobiology department at Imperial College London. And I was in the same year as Helen and both Elise in the STP. So started in 2019, and also like Helen, I got a job after in the department I trained in. So I've also been here for almost 5 years, which is a long time. Yeah, I did a BSc in computer science and then came straight to the STP in what was then clinical science, bioinformatics, physical sciences, what is now clinical science, clinical scientific computing, both mouthfuls.
Yeah, the training route's often convoluted, and we try and put loads in the names, don't we? Thanks very much. And Elise, you're in the same training year then as these guys. Whereabouts are you working?
So I'm currently working at University College London Hospitals Trust. Unlike Helen and Izzy, I did my training at Sheffield Teaching Hospitals. So I was there for 3 years in the scientific computing department, and then I moved down to London and I'm in the scientific computing department at UCLH. So my background prior, as I said earlier, was in maths. Really enjoyed maths, still have a great love for it. I ended up, after my BSc in maths, I did an MSc in mathematical medicine and biology. I really enjoyed the application of the coding that I did to medicine, and that's what kind of led me on to the STP.
Great, so quite a variety of different routes, albeit all coming through the STP. So I guess what would be interesting to know as well is what's the focus of the CSC activities in your department? How does it kind of work practically? Do you kind of do work for other departments? Do you just do work kind of for ones that you're kind of embedded next to? What's the kind of remit for you and your team? Let's go the other way around. Let's start with Elise this time.
Yeah, so having experience of two different departments has kind of highlighted how scientific computing departments can be very big based on the needs of the trust. So in Sheffield, it was more focused towards the medical physics side, whereas where I am currently in UCLH is more focused towards clinical engineering. So I work a lot with the clinical engineers. I'm actually for half of my time embedded in a biomedical engineering department, kind of helping with medical device integration with networking. And then I also do software development, particularly working with the ICU.
That's really interesting. And just to come back on one of those things, so your roles kind of split. Is that a formal role split, or is that kind of informally shared between those departments?
No, so it's more of a formal thing. It's quite like 50-50, essentially. I spend half of my time, it's actually at the National Neurology Hospital in the biomedical engineering department, and then half of my time sitting in scientific computing.
That's really interesting. Isadora, how about yourself?
So, as I said earlier, I am in the radiation physics department, and I am actually the first and currently only clinical computer scientist here. So, I think ideally the role would be shared a lot more around the hospital, but since there is just one of me, the majority of the work I do is for the radiotherapy physics department. I help commission software. I write my own software for use around the department, manage a couple of databases and servers, communicate with IT, and also advise a little on medical device regulations, as is such a big topic right now.
Right, so that's really interesting. So that's very much an embedded role where you're kind of, on your own in some ways, kind of supporting a specific kind of group. Does that present some challenges in terms of, obviously, if you're developing software, you obviously need some independence in terms of testing and things. Do you work with kind of the clinical scientists within radiotherapy then to do that, or do you have to kind of go to kind of other departments to be able to get that kind of help?
Yeah, definitely. There can be some issues with it. So I do most of the in-house software I've developed is for the radiotherapy department, and I am surrounded by physicists who are luckily quite keen to code. So either they help with testing or I test for them, stuff like that. But that's mainly within the treatment planning systems. So we use RayStation, so you can code within it in IronPython. And that is the majority of the coding they do. And then some of the stuff I do is outside of that. But yeah, testing can be an issue. I rely a lot on user testing and things like that.
And is part of your role kind of helping to upskill those kind of keen coders from the radiotherapy perspective, or is that not something that you do?
No, that's definitely part of my role. I have introduced, I'm just kind of finishing it up now, our first version control. So we're finally implementing GitHub, which while they were coding before I arrived, this is the first time they've used any sort of version control apart from, folders and typical radiotherapy quality management systems. So that's been really good. And yeah, I mean, they're all obviously very talented, but some haven't had formal scripting training. So it's a little bit of like, please don't do that. Let's do this instead.
Yeah, no, completely. I'm also from a radiotherapy background, so I completely can see the need for that. Helen, let's come to you. So in terms of your department, how does that look? What kind of activities do you support?
So the remit of our department is really quite broad. We're lucky to have really quite an extensive scientific computing team. We're up to about 25, 26 members at the moment, and we work closely alongside NCCPM as well, which is the National Coordinating Centre for the Physics of Mammography. So they are heavily involved in the breast screening, the National Breast Screening Programme. So we kind of coordinate with them a lot as well. So our scope is kind of to provide support to the medical physics department. We're placed within medical physics, and they have their different departments. So radiation protection, nuclear medicine, radiopharmacy, and radiotherapy physics. We also do a huge amount of relevant research and development. Again, a lot of that has a medical physics sort of bias to it. So a lot of it is about medical imaging. We have kind of dose monitoring software that we've developed, things like that. We also do take on sort of externally tendered projects that aren't research. So then we've got projects that are tendered by NHS England, and kind of we bid for those projects to take them on as a team. So as we get bigger and bigger, we're more able to sort of take on more of those kind of bigger national projects. And kind of my day-to-day work, most of it at the moment is on the system that we've developed ourselves to collect images from various hospitals around the country in order to use the data, the anonymized data for various different research projects. So we've got a kind of large collection of images, mostly mammography, but we're starting to move into other domains such as MRI as well. But the kind of the process of bringing those into our domain, anonymizing them, and pulling out what relevant data into our databases is quite a big part of my day job.
That's really interesting. So in some ways, it's the other end of the scale, isn't it?
Very much so, yeah.
Probably one of the biggest, you know, kind of scientific computing departments in the country. Really interesting. I suppose that whilst there are obvious massive benefits to that, are there any particular challenges that kind of come up as being part of a really big team?
Honestly, I've only ever experienced the benefits. We have a real range of different skills and expertise. So I found very much throughout my STP training, I was very well supported in different areas, lots of people that I could go to for advice and for training and things like that. So honestly, yes, it's only been a benefit to me, I think. And I think we are lucky with the people that we have in the team. I think perhaps one of the disadvantages that you could get would be kind of multiple different personality types and maybe some kind of people not getting on with each other as a team grows. But we've been really lucky in that aspect as well.
That's great. I mean, it sounds a really good place to work. I guess thinking through then, kind of like thinking back to when you kind of were thinking about going into the STP, is there anything that you wish that you'd known kind of before you started, or kind of as a trainee that you now know after 5 years in the kind of in the workplace, as it were, as qualified? Let's start with Isadora on this one.
Ooh, starting with me for the most difficult question. Nothing springs to mind. I would say that it's kind of on the point earlier about different size of departments, but the clinical scientific computing community as a whole is quite tight-knit and it's quite easy to find help. Like I've sent out emails and stuff to not just these two, but other people, and everyone's been really helpful. So I think probably the only thing I can think of is to make use of that community more.
That's both reassuring, I guess, that the trainees have a good experience in terms of the community, but yeah, good encouragement to for everyone to try and make use of that, kind of build that community. Elise, do you have anything that you think, oh, I wish I knew that?
I'd say, I don't know, probably more my approach maybe when I started the STP. I think I was really keen to kind of get things done rather than appreciate that I'm building a portfolio of work. So I think it's kind of important to consider what you're actually learning and your understanding of what's going to go forward, because that's what you're going to use, you know, as a qualified clinical scientist.
And Helen, anything to add on that?
I think kind of linked to what you've just said, Elise, was my kind of only thought with this one was wishing that I'd perhaps spent a little bit more time on the kind of theory of what I was implementing during my training. So kind of giving myself a bit more time for the greater foundation of mathematical techniques, statistical learning, that kind of thing. Because right now, most of what I'm able to do is sort of from an application based. So I'm able to understand enough to implement what I'm doing and to do that to a high level, but I always wish that I'd kind of more time to sort of study the real foundations of some of the work that I do now. I think, yeah, the SDP with the masters, so with the teaching that you get on machine learning and statistics and things like that, so using that as a sort of, oh yeah, I wish I'd used that a little bit more as a springboard for my own learning.
Yeah, I mean, I guess that's in some ways that's a good lesson for any specialism, isn't it? Is kind of a focus on the fundamentals to then be able to use that. I guess then kind of moving on from when you're a trainee, what's been the best things about your experiences so far in CSC over the last few years? Let's start with Elise this time.
So I would say for me, particularly in the role I'm in at the moment, it's very multidisciplinary. So I'm often working with engineers, people from IT, clinicians, you know, both doctors and other clinical scientists. And it's been really good to see different people's approaches to things. And it's definitely something I found valuable, particularly, you know, in terms of coming in as someone from scientific computing, perhaps understanding that not everybody understands different words or different concepts and kind of appreciating how to communicate.
Yeah, Helen, do you want to say your greatest experiences?
Yeah, I get similar. So the moving from maybe sort of doing some smaller projects during the training that can be quite just on your own, developing something that somebody's asked you to do, to kind of getting involved in bigger projects that involve more of the team and other teams from other departments. I think that's been really, really interesting and rewarding to kind of get that sort of teamwork element in. I think one of the best things that I've done as well is I was able to be involved in the Topol Fellowship. So I kind of was able to take the master's project that I was doing as part of the SDP further. So the fellowship gave me a day and a half protected time to carry on working on that research project. And I think that was really nice to be able to have that continuation of some research that I was really interested in as well. And then I think kind of the main things that I love about my job that now, I mean, similar to the training as well, but there's always new things to learn. I'm kind of never doing the same thing day to day. And the fact that my job is just lots and lots of problem solving tasks, which is exactly what I like to do.
That's great. That's great. Isidora, is it similar for you?
Yeah, I'd say both Helen and Elise's answers both apply to me. The only thing I can think to add is, again, potentially partially because I am in a small department, but very much from day one, it's been like I've been trusted to do things, and my opinion and experience has been taken very seriously. And I think that's partially because clinical computer scientists are a reasonably small field, so you kind of have to take them seriously. But yeah, just lots of different problems. I use a lot of different tech and software and languages in my day-to-day life, and I've got a lot of autonomy over what I do.
That's great. That's great. We'll finish with one last question just for the last few minutes. And this is perhaps a more difficult one, is do you think from your kind of initial experiences, is there anything we need to change about how CSC departments work, how work is managed, how kind of the NHS interact with kind of clinical computing? The answer might be different for different ones of you, I guess, because your context is very different. But I'd be really interested to know if you have any kind of thoughts on that. Let's start with Helen this time.
So I think just bouncing off what something that Izzy mentioned earlier, which was the kind of the real benefits of the network that you gain as part of the STP. I feel like following through with that to a greater extent, so more collaboration between departments. I think we can end up feeling like we're developing by ourselves, and perhaps other departments have done similar things that we could learn from, or kind of just sharing our knowledge between our different teams, I think is something that we could maybe do a bit better. I think we're trying to, we've got a network for clinical scientific computing to kind of help with that sort of knowledge sharing while we train up new trainees. But I think as well, for the kind of day-to-day stuff that we do as clinical computer scientists, a bit more collaboration between us would be really good to see.
Yeah, Elise, have you got extra things to add there?
Yeah, I mean, I kind of echo what Helen said, really. It's because it's such a small field, maybe people don't necessarily know about us to come to us to help with problems that we would be able to give kind of input into. I'd say the only other thing I would add is, in terms of the software development side, is kind of how people develop within the NHS is very different depending on the department. So you could see from the first presentation, obviously that seemed very kind of structured and they had a good framework in place. But for a lot of people, say, it's solo development. So I think it'd be kind of good to have teaching about the different ways people can develop and good structures for that.
Yeah, really interesting. It's very difficult, isn't it, when there are so many different contexts to where people are doing this kind of work. And Isidora, what would you add to that kind of?
I mean, I agree with both of those again, and apart from sort of more publicity might be the wrong word, education around what clinical scientific computing is. The only other thing I can think, and it's not really anyone here that can do anything, it's more the government, but more transparency around medical device regulations would be great, because it's very confusing, especially re-software. And I think that kind of built on what Elise said about a more NHS-wide process for software development would be great.
That's really interesting. Some really good points. It'd be nice to be able to expand a bit more on them, but I think we're out of time now. So a big thanks to all the people on the panel and all the speakers we've had today. It's really great that people are willing to put themselves forward and to present their work, which I think is often those who are early in the career are doing the most interesting and exciting work. And so it's great to be able to see that. A couple of upcoming events just to briefly tell you about before we finish. I think I'll probably come up on a slide in a moment. So we've got another webinar coming up in September, which will be around kind of how to talk IT, kind of some of the ways that we might interact with the wider IT team, and kind of thinking around some of those topics. There's also the IPEM kind of STEF conference coming up. And the abstract deadline for that is very soon. So do have a look at that. And then in terms of another CSC specific event, there's the one-day symposium in person, which will be in November. So do pencil that into your diaries. Thanks very much, everyone, for attending today, and good to see you again in the future.
Yeah, okay.